This is Chapter 16 in AP Chemistry, Chemical Kinetics, the Rates of Chemical Reactions. This will be the first of several screencasts from this chapter. You're probably very familiar with the indicator phenolphthalein. Phenolphthalein, as you might recall from labs having to do with titrations of acids and bases, turns a clear solution pinkish when you get into basic range, somewhere between pH of 8 to pH 9. If you're doing a strong acid, strong base titration, there's such a rapid descent, we can even use it to find the equivalence point at a pH of 7 because it shoots up so rapidly. In any case, what you probably didn't know, and I had forgotten, is that if you keep adding base to this solution, instead of it getting darker and darker pinkish or purple, it actually begins to fade until it becomes clear again, just like it did with a pH less than 7. So what's going on there? Here we are adding hydroxide ions, maybe sodium hydroxide or potassium hydroxide or something. And all of a sudden, instead of the pink color at least persisting, if not getting darker, the solution begins to go clear. What's going on? Well, here it helps to know something about the phenolphthalein molecule, about its structure. And here we're addressing one key concept in this chapter, and that is reaction mechanism. What is actually going on when the particles collide with each other during the course of a chemical reaction? And so here you see on the left a molecule of phenolphthalein with its 3 benzene rings. We have an oxygen ion there. There might have been a hydrogen at one point attached to it. Here we have a carbonyl. Over here we have an acetate. There's a C, and then there could have been a hydrogen there earlier on. And now as we add hydroxides at a certain point, when this molecule here, which is already red because of the fact that, or pink, whatever you want to call it, because of the fact that it's donated those protons to the base that we've added, at this point, there are no more phenolphthalein molecules left to donate those protons there and there. And so what happens then, the hydroxide actually adds right there. And in that process, this double bond up here becomes a single bond. And in a sense, what we're doing is we're removing hydroxides. The hydroxides that we're adding at this point, past the end of the available phenolphthalein molecules for donating protons in the color change from clear to pink, those hydroxides actually become part of this phenolphthalein complex, if you want to call it that. So in a sense, we're removing hydroxides from solution. And what happens when we remove hydroxides from solution? The solution goes back to being clear again. So you can remove hydroxides by adding acid, or you can remove hydroxides in this fashion right here. So there are 2 key ideas in this chapter, very, very important. Key idea number 1 is about chemical kinetics. Kinetics, of course, refers to the equation, if we talk about kinetic energy, of 1/2 mass times velocity squared. So chemical kinetics is about the particles moving fast, having a velocity. But even more so, chemical kinetics is about the reaction itself, taking time. Some reactions take place instantaneously, like an explosion. Other reactions take a long, long time, even years. For example, the rusting of something or oxidation of something in a very dry, desert-like environment. And of course, anywhere in between. So if we're going to understand the rate of a chemical reaction, rate is going to be something divided by time. We have to understand We have to be able to put a stopwatch to this or a timing device to see how long it takes for the reaction to go from products to reactants. It might go all the way to completion, like with a precipitate, a gas, or a molecule like water with a strong bond, or it might reach equilibrium. In either case, this can be timed so we can find the rate of the reaction. The other thing is we're very interested in reaction mechanism. What exactly is going on at the macroscopic and then the particulate level? What are these molecules, ions, or atoms actually doing? What's colliding with what? Now, wouldn't it be cool if we had a super-duper microscope that could actually film the collision of the particles so that we could see what's going on? And indeed, we're getting closer to that. And I would imagine that at some point, as our photography improves, even though the images might not be terribly clear, if we feed information into an artificial intelligence algorithm based upon what we know and what we might have clues about the kinds of particles that might be formed in between the reactants and the products, eventually we might be able to use a computer with the artificial intelligence to create a video. It would be a computer-generated video, not an actual video likely, of the particles, and we can see what they are actually doing so we can understand its mechanism. That is to come, and maybe you'll be involved in that. All right, rate. What do we mean by a rate? A rate is something divided by time. Sometimes we use the word speed. Speed is going to be, for an automobile, how many miles per hour. Miles divided by time, or kilometers per hour. If we are talking about the rate of flow of water through a faucet or a hose or out the spigot in the laboratory, we would be measuring the volume of that water, maybe in liters or quarts or gallons or something like that per period of time. In any case, rates are going to be something divided by time. And we can simplify this expression here in words by using the delta symbol, which is the Greek letter D. And delta means change, so we can have the change in concentration. And I'll use the brackets, which means molarity. That's something you better remember. Molarity is often represented by brackets, the concentration of one of our reactants or the concentration of one of the products, and we're going to divide this by the change in time. So that's another way to write the rate of reaction. And again, how are we going to measure the rate of this reaction? Well, we might measure the rate at which a reactant is consumed, or we might measure the rate in which a product is formed. It all depends upon what the reactant or the product happens to be. Some things are easier to measure a change in than others. And we're going to use devices, maybe a pH meter, maybe a spectrophotometer or colorimeter for measuring the change in the concentration. If we look at this picture right here, we have some blue food dye that's being added to water. So we have a blue water solution, and then we add bleach. So here's the bleach coming down. And as we add the bleach, a reaction takes place. Bleach is sodium hypochlorite. Sodium hypochlorite. And you'll notice that the color fades, or to use a play on words, the color gets bleached. And later on, it is even clearer yet. Over here, it's even clearer. Now, in this case, we're starting at time equals 0. So we start the stop clock, and then you can see at 2 minutes and 30 seconds, it's faded a little bit more. And then after 5 minutes, it's faded a lot, and maybe it stops and doesn't get any clearer than that. In any case, we can take samples of these water, each water, put it in a cuvette, put the cuvette in the spectrophotometer, or in your case, a colorimeter, and we can measure this concentration using a colorimeter. In fact, you're going to be doing this, if you haven't already, in lab, I think it is lab 25. But if you haven't done that lab, you will be doing a lab on the decomposition of hydrogen peroxide. I'm talking about the wrong lab here, so I'm going to erase that. We can also use a pH meter, like they say here, if there's a pH change as a result of this reaction going from left to right. So here's a particular example. Let's say we have dinitrogen pentoxide that decomposes into nitrogen dioxide and oxygen gas. Notice the stoichiometry is a 1 to 2 to 1/2 ratio. Now, if we were to graph this, the change in concentration, which means we'd have to be taking samples of the reaction at various points in time and testing their concentration, we could get a graph that you see here in blue. Now notice it doesn't go a straight line. It's not linear. It doesn't go straight to zero reactants. We might ask, why is that? Why doesn't it go straight to zero? Well, I'm going to erase that line because it doesn't do that, and answer the question. Or you should pause this screencast for a moment and see if you can answer the question before I continue. So, when we are at the very beginning of the reaction, we have lots and lots of reactants by comparison to what we have later on. So if you have lots and lots of particles of reactants, you're going to get lots and lots of collisions. And so the reaction is going to be fast, and you're going to have a steep slope. But then halfway down this curve, you don't have quite as many particles. They're more spread out because the particles you used to have, many of them have decomposed or chemically reacted. They're not there anymore. So now you have fewer particles, and the reaction has a more shallow slope. It's slower. And then when we get down here, you have hardly any reactant particles left at all. Every now and then they might collide with each other, and so you have the shallowest slope of all. Not a very fast reaction. Remember, the slope in this curve is the rate. The change in concentration divided by the change in time. The slope is the rate because you take your rise, molarity, over your run, time, which is the definition of rate in reaction chemical kinetics. Now think about an analogy. Let's say you go to Six Flags where they have those bumper cars. I don't know if they still do. I haven't been there in decades. But imagine you have lots and lots of cars out on the track that they're running on. You're going to get lots and lots of collisions. But imagine that every time two cars collide, they have to leave the track. Or maybe the cars break down, in which case they're removed from the track. This means you have fewer and fewer and fewer cars on the track, and that means you're going to have fewer and fewer collisions. The rate of collisions will get less and less. You have way more accidents in traffic than you have on wide open and untrafficked highways. Now, the mathematics that's done here are spread out over these two pages. This first calculation right up here is talked about at the top of the next page. And then we have these other reactions are at other places on these other pages. The point being that there are two strategies here. I'm going to erase some things so we can see it a little better. Let's imagine that we want to find an average rate. Well, in order to find an average rate, we need to pick 2 points. Like we might pick from here to there, or we might pick from there to there, in which case we need to find my molarity there, which you can see is 1.40, and my molarity there, which you can see is 1.10. And then and I have to subtract to get my change in molarity. And then I come down here to find my change in time, which is 1 minute. And that way I can find my average rate of dinitrogen pentoxide decomposing over this part of the curve. We can do the same thing down here. Now, what if I want to find an instantaneous rate at this particular moment? I would have to make a tangent to the curve, and it would be better and more accurate if a computer generated that than rather me trying to eyeball it as best I can. And then I would take the rise over the run, and that rise over the run would give me the instantaneous rate at that particular point. Because the tangent, remember, these points are at the end of the tangent. They're not on the curve. Now, better yet, you could use the methods of calculus, and if you knew the equation of this curve, it's probably an exponential, perhaps. If you knew the equation, you could take the first derivative of that, and the first derivative would be the rate at this particular point. So you can see that these calculations are fleshed out in greater detail. And by the way, let's mention a few other important points. Number one, you'll notice the negative sign right there, positive sign, positive sign. That's because your reactants are being used up. They're becoming less. And product number one and product number two are increasing. So you're going to get more and more of those. Another thing that you would be able to see is that if I can calculate this rate right here, then the rate of this is going to be half as much. And the rate of the production of this is going to be twice as much based upon the stoichiometric ratios. And then this instantaneous one is calculated down here, and we've already talked about that. If we look at another example problem, and this particular compound is called phosphane, phosphorus trihydride, or phosphane, decomposes into one form of phosphor, which is 4 atoms connected together. So instead of being diatomic, it is technically tetraatomic or quadraatomic. And then you also get, besides phosphorus, you get hydrogens in a 4:1:6 ratio. And if we knew the rate of decomposition of my phosphane right here, this rate, the rate of the formation of phosphorus, would be 1/4 as much, because you can see from the stoichiometry it's a 1:4 ratio. And you can also see that the formation of phosphor would be 1/6 the speed of the formation of hydrogen, because we have a 1:6 ratio of products. Coming over here, again, we can find either the average rate by picking 2 points. Let's say we want to go from there to there. I can find my average rate by knowing that molarity and by knowing that molarity. And that change in molarity goes in the numerator, and in the denominator, you have 2 minutes. My change in time is 2 minutes. And I can find my average rate during the first part of this decomposition reaction. If I wanted to find the instantaneous, again, I'd have to draw a tangent to the curve, or I could use the first derivative of the equation for the curve. Now, if you haven't seen it already, you will see a demonstration with the iodine clock reaction. In fact, not just the iodine clock reaction, but you're going to see a better variation of it called the Nassau clock reaction, Nassau, which happened to be the name of the administration building at Princeton University, where a couple of teachers one summer were able to reproduce the colors of the school colors of Princeton in a clock reaction. Now, by clock reaction, it means we can clock this. It's not instantaneous, nor is it super, super slow, though we could make it slower. But we can put a clock on it, and we can make it go fast or make it go slow, depending upon varying two things: varying the concentration of the reactants or varying the temperature of the reaction. By varying the concentration, if you increase it, you get more collisions, it goes faster. If you dilute your solutions, you get fewer collisions, it slows down and takes longer. You can also change the temperature. Warmer temperature, particles move faster, and a colder temperature, the particles move more slowly, and so the reaction slows down. And if you think about it, just a theological basis to go all the way back to Genesis 1, verses 26 and 27 and 28, you can see here that part of ruling and subduing God's creation, and by that I mean we're doing it the way God commands us to do it, using stewardship. He wants us to be able to exercise dominion over his creation. He's put us as the park rangers in charge. And one of the ways of having dominion over creation is to control chemical reactions, one of millions of responsibilities we have within the environment. We can control this clock reaction this way. And of course, even non-Christians want to do this because some reactions are just too slow and you want to speed them up. And other reactions are too fast and you want to be able to slow them down. That's very important, especially in medicine. So, in the Nassau clock reaction, you get a change in color that is very dramatic and very sudden. And what we know of the process is summarized here, but if we come down to the actual pictures, when you combine starch, regular old potato starch or some other starch, plus iodine, you get this purple complex, this purplish black complex. So this beaker actually starts out perfectly clear. I should say the solution starts off perfectly clear. And then all of a sudden, it flashes black or blackish purple. And that's because things have to happen. Molecules have to collide, and eventually you make the starch-iodine reaction or complex, and the reaction stops. Now, in the Nassau clock reaction, you also get something else that happens that's quite spectacular and gives us some more information. And hopefully you'll see a demonstration, if you haven't already, to know what I'm talking about. By the way, you may have done this actual test way back in grade school or middle school, or maybe in biology, and that's the starch test, which means you have a whole bunch of foods, fruits, vegetables, and other kinds of foods, and you have a bottle with an eyedropper in it filled with some iodine. And as you put drops of iodine, if it turns purple, you know there's starch in that food. So the vegetables and the fruits will not turn purple, and the meat won't turn purple. But if you have pasta and potato or rice or bread, those foods will turn purple because the iodine will complex with the starch. Now, in these pictures, they change some variables. The first thing that they do from picture A to picture B is they do the same chemical reaction, but this time they dilute, they add some water. And when they add the water, instead of the reaction taking 51 seconds, it now takes about a minute and a half. It slows it down. You don't have as many collisions when you don't have the same concentration. Notice that the level of the liquid is higher because of the additional water. Then in the last picture, we increase the temperature. We make it hotter. So we turn up the knob here of the heat, and it's hotter. The other knob probably is doing automatic stirring for us. And now the reaction goes very fast. Notice we're using the same quantity as before when the dilute solutions when we add them together, but notice how much faster the reaction is. It's back down to under a minute. Now we're going to pause here for a little bit because there's a lot to learn. First of all, another way to speed up a chemical reaction is by adding a catalyst. And if you get asked this on a test, and that's a strong likelihood, you should have two parts to your answer to the question, what is a catalyst? The catalyst is going to be some substance, a chemical, a compound that speeds up the reaction, making the reaction go faster, but remarkably, surprisingly, it's not used up in the process. It is not a reactant. It is not a product. In fact, you can use it over and over and over again if it's possible to collect it from your resulting solution. And you've heard of catalytic converters that catalyze the decomposition of nitrous oxides, NXOs, various nitrogen oxide compounds that form at the very, very high combustion temperatures of modern combustion engine automobiles. Since the atmosphere is like 79% nitrogen, you're going to get these nitrogen compounds, and you don't want them in the atmosphere, so your catalytic converter helps to break them down. And you use that catalyst over and over and over again. Unless, of course, somebody comes along and steals your catalytic converter, which is happening way too frequently, periodically in various cities. Now, in the first picture here, you can see a container of hydrogen peroxide. It's 30%, which is way, way high and kind of dangerous. You would not have that in your house. You probably have 3% hydrogen peroxide in your house as a topical thing to put on something that might be infected or to sterilize a wound or a scrape or something. In any case, if you pour some hydrogen peroxide into this beaker, it's going to sit there and slowly decompose inside a closed container. It might stay there for months or even years and still be active. But as soon as you add a catalyst, and by catalyst, I mean, for example, I could add some finely ground up iron filings from, say, a rusty nail or something. And all of a sudden, with those particles creating surface area for chemical reactions to act on, the reaction will take place within a fraction or two of a second. Just all of a sudden, it'll produce all of this steam. It's exothermic, and you're going to be getting from decomposition oxygen and water, and the oxygen is what is escaping, and some steam. Now, so the thing over here in this picture, we have an enzyme catalyst. Biological catalysts are called enzymes. You probably remember that from biology class. And in your body right now, for example, you have probably at least 2,000 chemical reactions going on, generally involved with cellular respiration and digestion and things like that. And every one of those reactions, being organic reactions, have a tendency to be on the slow side. So they all have corresponding enzymes that facilitate or make them go at just the right speed, like clockwork. You get a fever, and now the reaction rates are not going like clockwork anymore. And now they're not taking place in the right sequence at the right time, and that's why fevers are so dangerous. I could say another catalyst that they used over here is manganese dioxide. That's inorganic. Enzymes, of course, are organic. and enzymes are proteins. So they are very big, complicated molecules, which helps to explain why organic reactions are often kind of slow and need to be sped up sometimes. So that's all very interesting, and it's a huge field of research because we're always looking for new and better catalysts. Sometimes we have reactions and there's no known catalyst to make that reaction go faster. Sometimes the catalyst we're using is very expensive, or maybe it's toxic, or maybe it's not good on the environment to try to dispose of it. So we're always looking for better catalysts. Now the middle picture is really remarkable. And in this middle picture, you have the bombardier beetle, and it's got this little turret at the back of its abdomen, which can squirt out the same steam that you see over there. And it can direct it in a 360-degree angle wherever it wants to when it sees, for example, a danger or a predator or something like that. And in the abdomen, you have two sacs. One sac contains the catalyst, and the other sac contains hydrogen peroxide, which is normally kind of toxic to cells. So it's got to be in a protective container. And the bombardier beetle, using instinct, whatever that is able to combine these two chemicals together, creating the chemical reaction where it can shoot out the steam at a predator. Now, where does instinct come from? We're going off on a tangent here. Animals, all kinds of animals have instinct. They seem to have this knowledge. Sometimes this knowledge they learn from their parents. But oftentimes, way often, they seem to be born with this knowledge. And is this knowledge coded on the DNA? Well, to whatever extent it is coded on in their DNA, it's always been there. Where did it come from? You can't imagine a slow, gradual process over time where it gets this protein, then that protein, then that protein, because all along the way, it does not have the defense mechanism because it doesn't work yet. It doesn't have all the parts. And all of these unuseful parts, this partially formed spray mechanism and these partially formed reservoirs and so forth, and these partially coded for catalysts and hydrogen peroxide, all of this is a liability. It's a drain on resources. There's no macroevolutionary explanation that can take us from a beetle that has no mechanism like this to one that does. Now, this whole field, by the way, of studying animals and plants and learning from them about these nanomachines or these complex mechanisms and seeing if we can take advantage of that and use them ourselves is called biomimetics. Or sometimes it's called biomimicry. And this is a big field of science in the last couple of decades. And that is to go searching in plants and animals for these amazing machines and devices and strategies and see if we can copy them and then apply them to particular problems or challenges or engineering things that we want to accomplish. Biomimetics. This is all another example of what is called looking for intelligent design, ID. And of course, as Christians, we would know who that intelligent designer is, Jesus himself, speaking as God the Son into creation, this intelligence. John 1:1, in the beginning was the Word, the logos, the reason, the rationality, the logic, just so you know what that word looks like in the Hebrew. I'm sorry, the Greek. In the Greek, in the beginning was the Word, the logos, from which we get the word logic. And these devices that I just described in the bombardier beetle would be an example of something that is irreducibly complex. By that I mean, if you remove any part, or if any part is defective, if any molecule or part hasn't evolved yet if that were believed to be the way it came into being. The whole thing would not work. And not only would it be dead weight, but it could actually be a liability for the organism because it doesn't have a mechanism for defense. The hydrogen peroxide could be toxic to the organism and kill it. It's certainly not going to live long enough to pass its genes on to the next generation. So now that you've got the two parts to a definition of a catalyst, and you know that biological catalysts are called enzymes, another way to speed up a chemical reaction is to increase the surface area. If you have some solids, to grind them up very, very fine. And farmers know about the danger of this. If I try to draw a farmhouse here, okay, so this is supposed to be a barn, okay, with a big door. And next to the barn, we have a tall silo. Okay, there's my barn. And of course, in the silo, you're going to have a conveyor belt that you're probably going to put all your grain or oats or barley or rye or wheat or corn in there. And that's going to stir up all kinds of dust and all kinds of, a lot of that dust is going to be the outer skin, if you will, of the grains and the corn kernels and the husks and things like that. And a spark can ignite that. Because these particles in here are so small, there's a lot of surface area that can come in contact with the oxygen in the air, and that can be one big explosion. And it is tragic that people who work on farms have been killed. Probably happens yearly somewhere from explosions in these barns. And so the goal, of course, is to reduce sparks or to provide a way for any spark to not go through the air, but to go like maybe perhaps through a wire into the ground. And I forgot to draw the little window at the top of the barn. Okay. The effect of concentration on reaction rate. And we're going to pick up and talk about this in the next screencast.